Welcome to a presentation of the tumor normal analysis submission to the 2014 Genomics England Bake-Off by NextCode Health. The following is a demonstration of how to arrive at the conclusions presented by NextCode in the Clinical Sequence Analyzer and Sequence Miner. Please note that the tumor mutation analyzer is presently in development, and the user interface will eventually be similar to that in the CSA. From the NextCode website, please log on using your email and password. Under the Studies tab, you will see a list of the studies available. We will begin with Study 7. Click on Open Sequence Miner to get Sequence Miner started. We were given the complete genomic variant file, so we don't have the sequence alignment to view, and we ran a specialized analysis of the variant. So to do this, open Sequence Miner, then open the Report Builder tab. Under the Report apps, go to Somatic Cancer Genetic Analysis. Under VEP Consequence, we want to select high and moderate consequence variants. So select the top one, hold down the Shift key, and select the bottom one. Right-click and click on Set Selected, and then Apply to select them all. We're going to use the default in this Report Builder. So click on Create Report, and the analysis will begin. The analysis will take a few seconds. So the analysis has given us 2,300 variants. We want to look at just somatic variants, so we'll click on the somatic perspective. Now this reduces the number of variants down to 641. Now what we want to do is focus just on variants in cancer genes. So we'll click on the cancer actionable pathways. And now we see that there are two variants in the sample that are in cancer actionable genes. The two genes are mTOR and AKT3. If we look at the variant type, what we see is that mTOR is a missense variant and AKT3 is a spliced donor variant. Looking at where the variant occurs in the protein, we see that the mTOR variant is in the kinase domain, so it's likely a loss of function mutation. So if we look now at the domain it's in, so the loss of function in the AKT3 is likely not pathogenic. The loss of function in the mTOR occurring in the kinase domain is likely pathogenic. So then we reported on the mTOR variant. For this case, there is a complete genetic sequence. We don't have the sequence reads to examine, so we have to leave the case at this point. So to go to Case 8, open the case in Clinical Sequence Analyzer. Case 8 is a male patient with kidney cancer. We analyzed paired tumor and normal sequence files. This opens the genome browser. To do the analysis, we have to open up Report Builder. We want to select the Tumor Mutation Analysis app. Now we're going to look for variants that are of VEP high and moderate impact. So click on the first variant, hold down Shift, click on the last VEP category you want. Right-click on this, select to select all the high and moderate variants. Click on Apply to apply this. We're going to run this app using the default. So click on Create Report to get the analysis started. So this now gives us the analysis of all the Genome England cancer cases. We want to right-click on Patient and select Patient 8, and then Apply. Now we're looking at the 1,500 variants that are in Sample 8. If we click on Filtered to see just the high-quality variants, you see here we're looking at 52 variants. If we look at only variants in cancer genes, click on the Cancer Genes Filtered perspective. Now we're looking at 5 variants. You click on the Record View button to see the table view. These genes, we can see that 2 of these are mutations in functional domains. The FOXO3 gene has a mutation in the DNA binding domain, so we're likely looking at a loss of function mutation here. None of these genes are, however, actionable, but the most interesting gene we found was the mutation in the FOXO3. We can verify the FOXO3 mutation by going to the genome browser, click on the Synchronize button here, and we'll move to the genome browser and open up this variant. Now we're looking here just at the normal sequence file. To open up the tumor file, we have to click on Source and then click on BAM, and then drag the corresponding sequence file, sample 44, over. And we see the variant. We can double-click to see all the reads with the variant. And if you follow the green line down, you can see the G variant in a number of reads. So it looks like it's a real variant. We don't see it in the control sample, the normal paired sample for this tumor. That's the analysis of case 8. Genome England case 9 is a male patient with kidney cancer. We analyzed a tumor normal pair. So to analyze this case, we'll click on Genome England 9 and CSA, open the study in Sequence Miner. Now to run the analysis, we'll start by opening a Report Builder tab. We want to select the Tumor Mutation Analysis app. All right, now we want to select variants that are high and moderate consequence. Select the first variant, hold down the Shift key, and click on the last best consequence line. Right-click, select all of these, and then click on Apply to activate these. We want to run this analysis using default parameters. Click on Create Report to begin the analysis. This gives us the analysis for all the Genome England cancer samples. We want to filter by patient, so right-click on the patient column header, click on Filter by Column, and then select Sample 9. Click on Apply. Now we see this sample has 11,000 variants. We want to just first filter by high-quality variants. So under Perspective, click on the filter. High-quality filter has reduced the variants to just 65 variants. And if we can click on the record view to switch to the table view. Now of these variants, only 8 of them are known cancer genes. So click on the cancer genes filtered perspective. Now if we look at these genes, active domains and actionable pathways. None of these genes are actionable. One of these genes is a frame shift, likely to be a significant mutation. This is a mutation in the PBRM1 gene. Let's look at this frame shift mutation in the genome browser. So click on this mutation, then click on the Synchronize button to open this location in the genome browser. Okay, this is opening it in the normal sample. We've got to drag over the BAM file for the tumor sample as well. At this point, you need to have, of course, BAM open in the file browser. So in green, you can see the deletion in this tumor sample, and we can double-click to see it in all the reads. This is clearly a high-quality variant, so we can confirm this in the tumor sample. If we look in the normal sample, we don't see it, and we can expand the view, and it's clear it's not present at all in the normal sample. Okay, so this is the PBRM1 variant in this tumor sample. We can also look in this sample for structural variation. To do this, we'd have to open up the VARScan2 folder and open up the events folder. So we open up VAR, and then we open cancer, and then VARScan2. And now what we want to do is look at the DNA copy events file. Double-click on this to open it as a tab in Sequence Miner. Now we want to look just for large-scale events. So let's go over to Size class. We'll right-click on the column. We'll filter to just give us large-scale events. Now we'll sort this by chromosome. What we see is a number of events in chromosome 12. And if we look across, we see that nearly all of chromosome 12 is duplicated. And we have a strong evidence of duplication for 12q and weaker evidence for other parts of chromosome 12. You see here the 1.2 and 1.3 fold copy number. It turns out that chromosome 12 duplications are common in kidney cancer, so we reported it as well. And chromosome 12 duplications are correlated with poor prognosis in kidney cancer. So it's interesting to report in this case. That's case 9, and we're going to go to case 10. Genome England case 10 is a male with cancer of the testis. For this case, we analyzed raw sequence from a tumor normal pair. We ran the sequence files through our pipeline. We did alignment, we did variant calling, and then we did the cancer tumor-specific analysis. To look at this case, let's open up case 10 in CSA, and then we'll open up the sequence reads this file in Sequence Miner. To start the analysis, we'll open up a Report Builder tab. We want to select the Tumor Mutation Analysis app. We want to look at variants that are annotated by Beth as high and moderate impact. So click on the first category, and then hold down the Shift key, click on the last category. Right-click and select all the categories that are high and moderate, then click on Apply to apply this. You want to run this analysis using the defaults for this app. Click on Generate Report to start the analysis. Now this has done the analysis for all the Genome England cancer samples. We want to just look at cancer sample 10, so we'll right-click on the patient, click on Filter, and we'll select patient 10. Click on Apply. Now we're looking at just variants in sample 10. This gives us 7,800 variants. We want to focus just on high-quality variants, so under Perspectives, click on Filtered. This gives us 20 total variants. We can click on the record view to look at this in a table. Now, we can look for known cancer genes in this list of genes with mutations. So under Perspectives, we click on Cancer Genes Filter. This gives us no genes. So for this case, we're not seeing any mutations in known cancer genes, which is surprising. Let's go back to the default high-quality filter. We can see that under the Gene tab, none of these genes are in actionable pathways. And we can also look at cancer gene lists and see that none of these are known cancer genes this way as well. So in this case, we have a number of variants found in this tumor with high confidence, but none of them are cancer genes, and more importantly, none of them are actionable. We can look for structural variants in this case. To do this, we need to go under Source, and then under var, click on the icon to open up the directory. Then we want to go under Cancer, and then we want to go under var scan 2 to look at the structural variance analysis. We open this up for case 10, click on DNA copy events, double click on that, and it'll open up as a tab in the Sequence Miner. Okay, this is giving us a large number of variants. Let's remove the limitation here so we're seeing all the events, and click on Execute to give us all of them. Now we just want to look at large-scale events. So under Size class, we'll right-click on the column header, click on Filter on the column, and just look at the large-scale events. Now we'll sort this by amplification. What we see is one region stands out. It's on chromosome 12. What we see is nearly 4 copies. And if we scroll over to the right, we can see that this is the entire arm chromosome 12p. We've got a duplication, in this case, of chromosome 12p. In testicular cancer, this isn't particularly prognostic, but it is quite interesting, and so we reported on it. We also did a follow-on analysis to look for a different kind of structural variation, looking for fusion events and translocation. Doing this, which I'm not showing here, requires a little bit of additional analysis. We found a fusion event occurring in the NUT1 gene, a translocation event between chromosome 5 and 15. NUT1 fusions are well known in cancer. They're associated with what's called midline cancer and indicate an aggressive form of the cancer. Common NUT fusion is a NUT fusion with a bromodomain gene. In this case, we see a fusion between NUT and a kinase domain gene, CSNK1G3, which is a gene in the Wnt signaling pathway. So we see here a fusion between NUT and a Wnt signaling pathway gene. which might be interesting for this cancer. We reported that as well. Okay, that's what I had.